Good morning, everybody.
Good morning, sir.
Good morning, sir.
Good morning,
Good
sir.
morning, sir.
Breakfast कर लिया मैडम? नहीं सर, अभी ऑनलाइन है क्या?
Good morning, Assistant Commissioner, sir.
Good morning, madam. Hello, good morning, madam. Are you audible?
Yes, sir. Yes, sir. You are audible, sir.
Hello.
Yes, sir. You are audible, sir. Yes.
Yeah, good morning.
Good morning, sir. Hello, Mo. Hello.
Good morning, sir.
Morning, teachers. Daniel, good morning. Am I audible?
Sir. Good morning, sir. Audible.
Good morning, sir.
Thank you for joining, sir.
Is my sound all right?
Sound is all right, sir.
But there is some echo. I'll just log out and then join again.
After speaking.
I'll put on camera as well as mute mat. Another program is going.
Yeah.
We have to put on the camera. So they unmute moderator. Unmute. Okay, na? Sit here.
మీరు కనిపిస్తారా నేను కనిపిస్తానా ఇది ఆన్ చేస్తే ఓకే వాళ్ళు ఇంకా సౌండ్ ఆన్ చేయనట్టున్నాను.
Good morning to all of you. Am I audible?
Yes, sir.
Respected officers, principals, and dear students, a small instruction is there. Please mute your audio because some disturbance is coming. All the participants are kindly requested to mute the audio throughout the program. Please mute the audio throughout the program.
ఇంటర్నెట్ నా డేటా వాడగలనా?
లేదు సార్.
Yeah, Daniel, am I audible now?
Very clear, sir.
Sir.
Okay. Subrat Garu, is my banner seen correctly?
Here you can see.
Next one.
None of that.
Yeah.
అది వీడియో మ్యూట్ అన్న చేయక నేను పర్లే
Again, we need to find out and then.
నాట్ ఏబుల్ టు హియర్ I'm not able to hear, sir.
Just 2 minutes. Madam is joining.
Okay, sir.
Shall we start? Yeah, madam. Shall we start? Yeah, audible, madam.
Where is my background?
Also, here it is difficult.
The video also is off.
It's on now. It's on. It was on earlier. Yes, sir.
Leave it.
Clean. Okay. Sir.
Where is it? I'm not able. Listen, I'm not.
You're not able.
Yes, sir.
Shall we start, madam?
Yes, sir. Yes, sir. Please, please.
Okay, thank you. Manjula.
Okay. Good morning, everyone. A warm welcome to this Regional Children's Science Congress 2022 in this inaugural ceremony. A warm welcome to our Honorable Deputy Commissioner Ma'am, Mrs. G. Anuswaya Ma'am, Regional Office, Hyderabad, as the Chief Guest of the program. Sir T. Gopala Krishna Sir, Deputy Commissioner and Director NLI Rangareddy as our guest of honor. Mr. S. Radhakrishnan Sir, AC, Assistant Commissioner, Regional Office, Hyderabad. Mrs. R. Usha, Assistant Commissioner, Regional Office, Hyderabad. Mr. Benny Joseph Sir, Assistant Commissioner, Regional Office, Hyderabad. Mr. K.V. Subbareddy Sir, Assistant Commissioner, Regional Office, Hyderabad. Other dignitaries of Regional Office Hyderabad, and our R. Daniel Ratnakumar Sir, Venu Vidyalaya, all the other respected principals of Hyderabad region, all the teachers, students of various Vidyalaya who have joined us today. A warm welcome to this inaugural program of 3-day long Regional Children's Science Congress 2020. Now, I request our Vidyalaya choir group, under the guidance of our music sir, Ramu sir, to present the Navodaya prayer so that we can commence our program. Choir group presenting the Navodaya song. Friends, everybody please stand up and join your hands. Thank you, students. Hami Navodaya Home is the inspiration and stimulation which keeps us going to every possible height and to aspire to achieve the unprecedented pinnacles of success. Now, as we all know that the main objective of Navodaya Vidyalaya scheme is to provide quality education to children predominantly from rural areas. NVS has been making a series of efforts for the quality improvement based on abilities and potentials of the children for creativity, innovation, research, and critical thinking. RCSC is one of the phenomenal steps in this line to inculcate scientific temper and promote scientific talents with the objectives of providing a forum for talented students, to help students meet eminent scientists and drive inspiration, to nurture and nourish creativity, to promote the opportunity for students to go beyond limits, and to prepare the young generation to face challenges by encouraging the development of experimental and investigative skills. Now, in this line, I will like to request our Honorable Assistant Commissioner, Sir, Mr. K.V. Subba Reddy, Sir, an embodiment of humility and grace, to present the welcome address, Sir. K.V. Subba Reddy, Sir.
Evening to everybody. Respected Chief Justice Srimati G. Ansuya Madam, Deputy Commissioner, Navodaya Vidyalaya Samiti, Regional Office, Hyderabad. Guest of honor, Sri T. Gopalakrishna Sir, Commissioner, Navodaya Leadership Institute, Rangareddy. Srimati Anuradha Madam, Assistant Commissioner, NVS RO, Hyderabad. Sri Radhakrishnan Sir, Assistant Commissioner, NVS RO, Hyderabad. Sri Benny Joseph Sir, Assistant Commissioner, NVS RO, Hyderabad. Sri Daniel Ratnakumar Sir, Principal, JNV Rangareddy. Principals of all JNVs, staff and students. NVS has been organizing the Regional Children Science Congress every year during the last 20 years. It is a unique program being conducted by Navodaya Vidyalaya Samiti. This year, this program is being conducted from 21st to 23rd February 2022. No other organization is conducting this type of program. This is a very, very important program. The main aim of the program is to provide a forum to students for creativity by watching and doing experiments, to help students to meet eminent scientists face to face, interact with them, and feel thrill of unique experience, to nurture creativity and innovativeness, to solve problems relating to their learning process, to promote opportunity, to go beyond school limits, visit labs of international standard, collect data, analyze and arrive at certain findings, to prepare young generation to face challenges by encouraging the development of experimental and investigative skills. This year, around 100 students are identified from 77 JNVs to participate in this unique program. The Regional Children Science Congress is being conducted through virtual mode this year because of COVID-19. In this program, the students are going to meet eminent scientists like Dr. Vivek, Dr. Banjari Kiran, Dr. Shashi Kiran, Department of Life Sciences, University of Hyderabad, Dr. Sarita Vag, Department of Indian Institute of Space and Technology, Trivandrum, Dr. Ashwini Dalal, Head Diagnostic Division, Center for DNA Fingerprinting and Diagnostics, Hyderabad, Dr. G. Manoj Kumar, Advanced Center for Research in High Energy Materials, University of Hyderabad, Dr. M. Hanumantarao, Advanced Center for Research in High Energy Materials, University of Hyderabad, Dr. Syed Nizamuddin, Department of Mechanical Engineering, IIT Hyderabad, Dr. Ranjit Ramudarai, Department of Material Science at Metallurgical Engineering, IIT Hyderabad, Professor V. Chandrasekhar, Tata Center Director, Tata Institute of Fundamental Research and Hyderabad, Dr. Sutapa Ghosh, Senior Principal Scientist, CSIR, Indian Institute of Chemical Technology, Hyderabad. Therefore, I request our Honorable Deputy Commissioner and welcome our Anusaya Madam, Deputy Commissioner Navodaya Vidyalaya in the Regional Office, Hyderabad, to be the chief guest of this inaugural session. And I also welcome Sri T. Gopalakrishna Sir, Deputy Commissioner Nallai Rangareddy, to this inaugural session, and all others to participate and make this program very successful and make this program very grand success. Thank you. All the best. Thank you very much.
Thank you so much, sir. Now, the person who is going to address us is a personality who can influence anyone with his communicable and contagious positivity and openness. The man of diligence and expertise, our guest of honor of the day, our Honorable Deputy Commissioner Sir and Director of NLI Rangareddy, Mr. Gopalakrishna Sir, to address the Assembly and grant us the pleasure with his erudite words. Respected T. Gopalakrishna Sir.
Thank you very much. Very good morning to all of you.
Good morning.
Am I audible? Manju, who is this? Am I audible?
Yes, sir. Yes, sir. Audible, sir.
Thank you. So, respected Madam Anasuya, Deputy Commissioner, NVSRO Hyderabad, Shri Radhakrishnan ji, Benny ji, Subbareddy ji, the Principal of the venue JNV, Daniel Ratnakumar, the staff of JNV Gachibowli, and the principals, teachers, and my dear children. It's a big moment of festivity for all of us, celebrating Regional Science Congress in the virtual mode. We have been very unique in the entire school education scenario of our country. During the pandemic, it was only Navodaya Vidyalaya Samiti who could contact and communicate with all the children who are held up at their homes with their e-content and with regular interaction of the teachers with the students. In a similar fashion, we have so many things to be mentioned as exclusive things that are being conducted only in Navodaya Vidyalaya Samiti, and Regional Science Congress is one such activities. Regional Science Congress is an activity which is longed for, which is awaited, which is always, with a lot of hope and excitement children look forward to every year. And this year, in fact, we wanted to conduct it in physical mode. Again, due to certain challenging circumstances, we could not do so. But still, the way JNV Gachibowli, under the able guidance of Shri Radhakrishnan ji and Subbareddy ji, who is the in charge of Regional Science Congress, has made wonderful arrangements. Partly, I was also part of it. For a few days, I was also looking at the program that is going to be arranged through virtual mode. It is an amazing program that you are all going to thoroughly enjoy. And, it's going to be a feast for all of you. And I just would like to appeal to my colleague principals and the other teachers who are present here. Regional Science Congress has been one of the best flagship initiatives of Navodaya Vidyalaya Samiti. I wish that this particular initiative, if it can be taken with the same vigor, with the same interest and same enthusiasm, how it is taken at national level, in the similar fashion if it can be taken at JNV level. Like, we plan it for the entire year, like, which are the schools going to participate and how we are going to do it, which is the venue and who are going to be our panelists and who are going to be the scientists and so on and so forth. With the same interest, with the same plan of structure, if the JNV can conduct it right from the beginning of the academic year, all the focus areas of National Education Policy 2020, whether it is experiential learning, child-centric learning, learning through joyful methods, learning for life, science for life, all that will be by default covered. So we have the tendency to make it as a standalone program. Like, our school is given the responsibility, so we do it. No, it is for the entire organization. It is for all the children. These children are already privileged. They're already ignited. They're already inspired. They're already on the fast track. We need the other children who are less motivated, who are less oriented, who are less introduced. Those children should be our target children, and those children should be reached out. And I am sure with the dynamic leadership of our principals, being the academic leader at school level, if Science Congress can be institutionalized to the extent that the planning is done right from the day one of the academic year, the children are made as partners in planning, the teachers are the architects of the planning, and with the concerted efforts of everybody, if it is executed, I'm sure this program can be further strengthened in our system, and it will be a wonderful opportunity for all the children to have the taste of learning while learning while participating in an experiment, listening to the scientists, listening to the fellow students. That is how I look at it. And a lot of arrangements have been made, and I am very happy that I have been invited to be part of this program. And as I'm sure you're all aware that with enhanced vigor and enthusiasm, we want our teachers to make use of the Atal Tinkering Labs, which have been set up with crores and crores of rupees of investment. Each item in the Atal Tinkering Lab is meant for the child to play with, child to meddle with, and child to work with, and child to learn science while doing. So I request all the teachers who are part of ATL labs to make use of the opportunities, and some of the schools are also part of Vigyan Jyoti, and with some exclusive programs are being organized, and all those programs, all those activities are available. The recorded versions are available. I request the science teachers to access them and pass on this benefit to not only girl children, it is for everyone. Learning is for everyone. So I request the principals to make I'm sorry, I think there was a small disruption of the network. So I take this opportunity to wish you all the best. It's going to be an amazing journey for 3 days, and each activity has been planned in order to inspire you, to motivate you, and to enlighten you, and to give you an opportunity to have hands-on experience on various activities. And I also thank Principal JNV Gachibowli and Madam Anusuya for giving me this opportunity to be part of this wonderful program. Thank you, one and all.
Thank you so much, sir, for your extensive and elaborative speech, sir. Definitely, it won't be a standalone program, and everybody will take part wonderfully. It will be a feast for the students. Now, I would like to request our Deputy Commissioner, ma'am, Hyderabad region, G. Anuswaya ma'am, who is an epitome of integrity and empathy, a true leader for her resilience, decisiveness, and goal setup. Kindly present the keynote, ma'am, and the chief guest address, ma'am.
Very good morning to one and all.
Good morning, ma'am.
Respected Deputy Commissioner Shri Gopalakrishna from NLA Rangareddy, my colleagues Mr. Subbareddy, Mr. Radhakrishna, Mr. Anuradha, and Mr. Benny Joseph, the host principal Mr. Daniel, and the principals of Hyderabad region, and dear students and other staff members. It gives me immense pleasure to be in this session of inauguration of Regional Children's Science Congress. You know that due to COVID pandemic, we missed last year's Regional Children's Science Congress. And this year, it was thought that if it is not possible or feasible to bring the children, at least in the physical mode, we should provide an opportunity to the students to be a part of the Regional Children's Science Congress and to motivate them into the field of science. And that's how now we are in this program. And you know, as Mr. Gopalakrishna had mentioned, NVS has taken up many flagship programs for the development of the science subject, the research, the inquisitiveness of the students in the field of science, and also to ensure that more and more number of students qualify in the STEM areas. And conduct of this sort of exhibitions is not new to NVS. Right from the inception, way back in 1989, we had been conducting science exhibitions at a cluster level, regional level, and national level. And conduct of this Regional Children's Science Congress had come in for the last 2 decades. And now you know that you are also part of the National Children's Science Congress, wherein your activities and your research papers are presented and you are going up to the national level. Students, it's a golden opportunity. It's a blessing in disguise. If it had been in a physical mode, only the selected students, a few selected students from each JNV would have got the opportunity of participating in the Regional Children's Science Congress. Now that it is on the online mode, every child of all JNVs can actively participate in this program and really get motivated. And I should be thankful to the coordinating and hosting JNV and the assistant commissioner that they have brought very, very eminent scientists, not only from Hyderabad, even from other places, science institutions, which is possible only because of this online mode. At this juncture, I welcome all the eminent scientists and guest lecturers who are going to be a part of this program for today and also for the days to come. Children, coming back to you, even though NVS is taking many initiatives to bring in the awareness of science and science researchers and making you to go into the field of science, it is for you to develop your interest, enthusiasm by actively participating in such activities. And not only this will not only be helpful for your academic improvement, it will also be giving you some view about science, and you may definitely get motivated to select science branches in your career in the higher studies. Students, as sir told that most of the schools are having the Untitled Tinkering laboratories, and it is a lab for your research, and you have to devise your time and you have to coordinate with your principal and teachers and utilize the time which is available and prepare yourself for researchers. See, I should recall here, if our scientists had not been more proactive, more involved and more dedicated, I don't think that we would have overcome this pandemic in such a short 2 years of time. Generally, any pandemic will go for a decade. Now that almost we have overcome that, and now it is going to be like a flu or a cold or something like that to a normal situation. which is possible only because of our scientists' contribution in the field of, development of this vaccinations in a very, very, very short period. And science is playing a vital role, not only in such medical areas, it is also playing in the researches of your defense and your development of our country, and taking our country's name into the top pedestal. And I wish and pray that our students should also become top scientists in future, and this science Children's Science Congress should lay a foundation for that, so that they will get motivated and they'll be becoming a future scientists of our nation, and more particularly they become the future scientists and they serve for our nation. That is more important. Children, you will have variety of activities now. Kindly, I request and appeal to the principals, allow the more and more number of students to be a part of this Regional Children's Science Congress, and let them listen to the guest lectures, which are going to be very, very special and meticulously planned for the students' motivation, to increase their motivation level, and also the activities which are going to taken up, like paper presentation, quiz, and other science activities which are going to be demonstrated will be very, very useful to the students, and they will be definitely get interested, and they may jump into the field of science because of this, because we do not know what is going to be a small ray that will go into their mind and design their future career. I think that the Regional Science Congress is one such activity, and I wish all the students very best, and I wish them and I appeal to them to make best use of it, and appeal to principals to spare, allocate time to the students to watch this program according to the time slot. And thanks to the coordinating JNV for arranging elaborately for this program. Thank you one and all. Wish you all the best.
Thank you so much, ma'am. Thanks a lot. Now I would like to request our Principal Sir, R. Daniel Ratnakumar Sir, to present the vote of thanks for this program.
Most respected Deputy Commissioner, Madam Anushya, most respected Deputy Commissioner Yellanli Rangareddy, Sri Gopalakrishna Sir, our respected Assistant Commissioners Sri Radhakrishnan, Srimati Anuradha Madam, Benny Joseph Sir, and Subba Reddy Sir, and resource persons and scientists joined in the inaugural sessions, all the officers of NUS Hyderabad region, learned principals, and all my students joined in this great event. Before thanking you, once again, I welcome all of you on behalf of Regional Office and JNV Rangareddy for this Scientific Feast Regional Children's Science Congress. Regarding the events, schedules are sent to all the schools. We have done our best under the guidance of our officers to call, to invite great scientists and professors in and around Hyderabad as well as outside Telangana. It is a great opportunity to every one of us to be the part of this program. It's all happened because of the great leadership and the continuous motivation of our Deputy Commissioner Anushya Madam on one side and Gopalakrishna Sir on the other side. From both their suggestions, we have proceeded and we have come to this stage. And I take this opportunity to thank both the officers for the continuous support to host this event in an appropriate manner. Next, let me take this opportunity to thank our Assistant Commissioners, particularly Subba Reddy Sir. He calls me every day and more or less he come to the school frequently. Yesterday also he was there seeing the things and guiding us, and other assistant commissioners, Radhakrishna sir, Anuradha madam, and Benny Joseph sir, all are in touch with us, and they have continuously motivated us, because this online program, virtual mode, is new to us, organizing such a big event. But we are able to do it only because of their continuous support. And I take this opportunity to thank all the officers of NVS Hyderabad region. Next, let me take this opportunity to thank all the principals for their continuous support, as well as the students and other teachers. And with these words, I thank one and all. Thank you all.
Thank you so much, sir. Now we have among ourselves Dr. Vivek, Dr. Manjari Kiran, and Dr. Sashi Kiran from the Department of Life Sciences, University of Hyderabad. I would like to have the opportunity to give a brief description about our scientists and our professors. First of all, we have Dr. Vivek. He's from Department of Life Science at the University of Hyderabad. Research interest of Dr. Vivek are in trait discovery related to C4 photosynthesis, integration of omics data of knowledge, mining of genes and genome, annotation improvement, taxonomic and functional characterization of gut microbiota for nutrition and diseases, taxonomic functional characterization of plant root microbiota for sustainable agriculture, nutrigenomic for important crops, and recently, Sir has been a visiting scientist of ICRISAT, which was recently in news because of PM Modi's visit. Since 2010 to 15, Sir finished his postdoctoral fellowship in C4 Rice project in the International Rice Research Institute. In 2016, Sir became the research associate or guest faculty at JNU, Indraprastha University, New Delhi. And 17 to 18, Sir was in Ramalinga Swami Fellow, ICRISAT, Hyderabad. And presently he is working as the assistant professor of University of Hyderabad. A warm welcome, sir. Thank you so much for being here. Next in line, we have our Dr. Manjari Kiran ma'am. And ma'am's area of research is cancer genomics, RNA biology. Ma'am is a computational biologist. She uses publicly available biological data to address fundamental questions in biology and generate data-based models. She did her PhD with Professor H.A. Nagaranjam at CDFD Hyderabad. In her PhD, she proposed a novel dichotomy of highly connected proteins, that is global and local hubs, using human tissue-specific protein interaction network. After that, ma'am went on to work as research associate with Professor Anindya Datta, University of Virginia. There she identified a set of 8 long non-coding RNAs whose combined expression can predict survival in glioma patients. Her work on prostate cancer specific tRNA derived fragments as a prognostic marker and their role in prostate cancer metastasis was funded by Department of Defense, DOD, for 2 years of postdoctoral training. Apart from these, Madam has worked on several collaborative projects involving novel long non-coding RNAs in cancer and muscle differentiations. A warm welcome, Madam. Next in line, we have our Sashi Kiran sir, again from University of Hyderabad. Sir's area of research is protein ubiquitination and ubiquitination in cellular processes and disease, HPV-induced oncogenesis, and post-translational modification in cellular activities. Sir's lab is themed on ubiquitin signaling and genome editing. Sir is interested in identifying ubiquitinases crucial in important biological process and identify inhibitors against such ubiquitinases. They apply individual depletion strategy to members of ubiquitin prostome systems to identify crucial regulators of cellular processes and diseases. Warm welcome, sir. Now I request our Vivek Thakur, sir.
Yes, good morning. Am I audible?
Yes, sir. Audible.
Yeah. Thank you, organizers. I think it's wonderful to meet, at least virtually, such a huge number of students. I think a couple of years ago, before pandemic, I got a chance to interact with the teachers of Navodaya Vidyalayas. And it was a wonderful interaction. We had a lot of scientific discussions along with the teachers, but this time I'm meeting students. So yeah, it's a great feeling. And I hope you all are excited, right? Yes or no? All excited for this Science Congress, Children's Science Congress? Good. Okay, so let me share my slides and then we can proceed. Okay, can the administrators, can they confirm whether my screen is visible?
Visible, sir, visible.
Yes, sir, it's visible.
Yeah. So as Madam introduced us, we are three faculty colleagues from School of Life Sciences, University of Hyderabad. And we are going to, so on day one, we are going to talk about some recent trends in the biological research. And this session will be followed by two more sessions. Another scientist from Institute of Space and Technology from Trivandrum. She will talk about cosmic wonders, and another session, which is the last session for today, that will be by Dr. Ashwin Dalal, who will talk about some interesting areas of biology, which I'm anyway going to touch upon. Okay, so let me start. So students, before I share insights about things that we have planned to share, the recent trends, I thought I'll start with what you already know. And the question that I wanted to ask you, how our understanding of biology has progressed. And I think in the virtual mode, I can't hear you, but then I think you all will agree that in past couple of centuries, it has progressed tremendously, right? Huge lot of information is there, very interesting information, lot of biological questions, we know the answers. And that is what you study in textbook. So earlier, for example, earlier we used to think that we human have been created specially. We are much different from other organisms. But is it true? I think the research from Darwin and other scientists, they have proved that it is not true. And in fact, what is visible on my screen is nothing but it's a tree of life. We all are interconnected to each other. So that was a very important finding. Let me talk about one disease. So this is I don't know whether can you recognize it, but I think some of you might have faced these kind of symptoms. This is nothing but chickenpox. And earlier, if you talk to your parents or if your grandparents, they will tell that, hello, is there an echo? Yeah, no. So they will tell you that, no, this is not an infectious disease. They will tell you that this happens because of fury of some god or some goddess. That's what they used to think, but this has been proved that no, it is nothing but it is caused by a virus. Okay. Similarly, moving on, let's come to the plant kingdom, the very green world, which our eyes like a lot. And what you see here is nothing that plants, they have a kitchen. Okay, so on the screen, what is shown here is a section of a leaf and the internal structures. And this is the kitchen of the plant where they cook food. Although when I was a student like you, I didn't believe that there is a kitchen inside leaves. But then with some experiments conducted by our teachers and some textbook knowledge, then I came to know that no, indeed there is a kitchen. Okay, and in your textbook you must have conducted some experiments how we can ensure that indeed there is a kitchen. Some photosynthetes are being formed inside the leaf. Anyway, so this was just to connect with all. But let me, are there still major challenges for discovery, or whether all the discovery that has been already made? But I think if anyone is allowed to unmute, admin, because I can't see if there is someone has raised hand, so I can't see that.
Hello, Dr. Vivek, sir. Are you there, sir? Yeah, we think sir left the meet. Maybe some network issue or something. Yeah. May I request Dr. Shashikiran, sir?
It's me, Manjari here.
Manjari,
I
ma'am.
think
Good morning,
yeah, Dr. Vivek has lost the connection.
Yeah, ma'am.
So shall we wait for 1 minute or so?
Yeah, yes.
Yeah, let's wait for 1 minute. If
Yes.
he still could connect, then I'll
Sure,
start the
ma'am.
presentation.
Sure, ma'am.
Hello, am I audible?
Yes, sir. Yes, sir.
I'm
Audible,
so
sir.
sorry,
Yes,
there
Vivek,
was
you
a
are.
power failure and I did not anticipate this. Can I resume my presentation?
Yes, sir. Please, sir.
Okay. Yeah, let me share my slides. I'm so sorry. So these technical glitches.
Yeah, it happens, sir.
Are you able to see my screen? Can anyone confirm?
Yes, it is visible.
Yeah, thank you. So the question that I was asking, are there still major challenges for discovery? And then I'll quickly come to my point. So, well, one challenge that the entire mankind is facing, and that is this pandemic, right? And but thanks to these scientists and researchers, in just a year's time, they were able to design vaccines, and now several vaccines are already available in market or which is provided by government. Okay, and in fact some of the children, they might have also taken those vaccines. So this is just an example. This doesn't mean that there is just one such challenge. There are many challenges. For example, a very contrasting example that I cite here is that loss of biodiversity. Okay, this is another important challenge, and we may have to come out with better solutions to that, rather than so some ongoing efforts are already there, like conservation of trees and forest and wildlife, so on and so forth, but more efforts are needed, innovative efforts are needed. So this means a lot of advancements are there, but challenges are also there. Okay, now the next question that I want to ask is that if deeper insights into a problem, can it address those challenges? So what I mean is, let me start with again textbook knowledge. So the students of class 8th, 9th, 10th, they have been all taught about this nervous system, where so you can recognize that this is nothing but an internal structure of a brain, right? You also know that this organ brain is it contains one kind of cells. Typically, it looks like this, which is called a nerve cell, an axon, and dendrites, and all those things you can easily recognize, right? Now, if we further, when researchers, with the help of microscopes and various imaging techniques, and various biochemical techniques, when they further looked into, so for example, this panel shows that muscle fiber, which is connected with these nerve fibers, right? And then even going deeper inside, you can see that this is how it is connected. These are called synaptic endings. So connections between two neurons, that is also synaptic ending, or this is a typical end of a neuron where it interacts with the muscles. It gives certain commands and signals. That is what the muscle cell will follow. So what trend do you see? We are going more deeper and deeper, okay, and we are trying to understand it. And that is what I wanted to tell, is that this is one of the important trend that you will also come across today. It is that we are trying to go at the molecular level. We are trying to understand the interplay between various molecules, okay, how they are talking to each other, what are their roles, okay, how their level increases, how their level decreases, how they are talking to each other. So these molecules are very interesting. You can assume that characters. So you have characters in your storybooks, but they also play a very interesting role. If you really read about all these biomolecules, you will be amazed to know that how interesting they are. And anyway, you are already studying the role of these biomolecules, okay, and you are becoming more and more familiar. Okay, so this figure I deliberately kept that. So this is a further resolution of the same story that I just talked in my previous slide. So here, what the researchers are further looking into, what are the molecules which are secreted by at the synaptic ends, which are called neurotransmitters, how they interact with various components. So these components can be like transporters, they can be channels. Okay, so this means now scientists are able to find out several of such proteins, but still they are still learning how each and step takes place, starting from why these
Sir, you are not audible, sir. Vivek, sir. Students, please. Only. Okay. Yeah, Vivek, sir. Sir, you are not audible, sir. Leaving, maybe some network issue. Students, we'll wait for sir. Please be on mute. We'll wait for sir for a few minutes.
Let him join again.
Yeah.
Ma'am, excuse me, ma'am. Excuse me, ma'am.
Yes, ma'am.
Yeah, I just now called him. It seems he was unaware that he is out of the meeting. I mean, he got connected, but he was unaware of it. So he will rejoin.
Okay, okay, ma'am. Okay.
Yeah.
That is better because they can click. एक मिनट आप उधर ही चले जाइए, मैं बाद में देखूंगा.
Hello, admin, can you allow me to share my screen?
Yes, sir. 1 second. It's done, sir.
Okay. My screen is visible?
Yes, sir, it's visible.
I don't know which slide was, when I was disconnected, I was on this slide. Can anyone confirm?
No, Vivek.
This one?
This one.
Yeah, here.
Yeah. So yeah, I had not moved a lot. So yeah, so I was asking a question that I talked about molecules and activities. We need to go deeper to understand the molecular mechanisms. But what is controlling these molecules and their activities? So what I'm showing here in this through this photo is a remote control. So all I wanted to ask, what is controlling them? Is there a machinery which controls these molecules, their levels, their activities? And in fact, the answer is yes. We all carry genetic blueprint. Okay, what we call, you must have heard DNA, or you must have heard genome, or some of you might have heard chromosomes. So our genetic blueprint, if let me turn on the laser pointer, you see these codes, they look like characters, okay, A, T, C, G, but in fact, these A, T, C, G, they represent the certain molecules in our genetic code, right? So our genetic code is packed in the form of these funny looking structures, you may call chromatin or chromosome, okay? So this genetic blueprint contains instructions how these molecules will be formed, what will be their activity, when their activity will start, when their activity will stop, okay? So moving on, so this is next trend that I'm going to emphasize is that we also need to, apart from the molecules, we also need to dig or mine the genetic blueprint that we have. Okay, not only for human, I'm talking in general, all organisms contain this genetic blueprint. Okay, and this is the latest trend, maybe in last 1 decade or so, that our genetic blueprint is heavily being mined, okay, through various modern technologies. And today you are going to have three talks, okay, and they all will be one or the other aspect. One
Manjurani madam, you call him. That screen is not moving. Audio is also not moving.
Yes, I'll do that. He's reconnecting. Sorry for the inconvenience.
Yes, ma'am.
Again, my sincere apologies. This is happening for the first time. Maybe Microsoft Teams is not liking my session. I don't know whether you guys are liking or not. I'm so sorry. Can anyone allow me to share my slides?
Yes. 1 minute.
But I'm sure Microsoft Teams is not liking my session. Okay, I'll try to wind up very quickly. Otherwise, yeah, it is really annoying for you all. My screen is visible?
Yes, sir.
Yeah, so what I was conveying is that a second important trend, and that is the genetic blueprint that the organisms have that need to be mined, that need to be analyzed, that need to be looked into. And you are going to have 3 talks on that genetic blueprint. Okay, moving on. Another question. So where these molecules can be found, the molecules I have been talking about, like I talked about neurons, within neurons at the neural junctions, I talked about neurotransmitters. So how can you find those molecules, and how will you examine their activities? This is another two question that I want to know. Because of limitation of this mode, I'll have to also talk on your behalf. Okay, so let me pick one case. So for example, if I talk about brain, and if I want to study the activity of certain molecules which is in the brain, okay, what I need to do? Do I need to take a volunteer and do the surgery and using biopsy take out a part of the DNA? But I don't think anyone will like it. Even the surgeons will not like it. I mean, really tampering with it unless and until it is very important. Okay, because this is a destructive mode. So this means that we have limitations. We have more limitations when we work with humankind or human individuals, human volunteers, because we cannot go for such destructive ways to obtain the molecules that we want to examine, or to see what molecules are there in a particular part of brain. No, we cannot do that. Okay, so this is a serious limitation. So is there any alternate? So we are going to talk about some alternates in biology, and that is very fascinating area. So what alternates? So on my screen, what I'm showing here is that the upper figure that is a human brain and various parts of the human brain, whereas the lower one, that is it's already labeled, that is a rat brain. Okay, so here you see that this is how the rat brain looks like. And do you find any commonality? I think since the color has been labeled, so based on the color levels, we can find commonality. Cerebellum is there, cerebral cortex is there. The sizes, one change that we notice, that sizes are different, but then most of the structures are common between these two. Okay, maybe their activities, definitely they differ to some extent, but then lot of structures are common. Now, so given this, if we can't work on human subjects, human individuals, but can we work on an organism, for example, rat and mouse, can we work on that, and can we use their brain as a model to do some research, do some discovery? That is what I wanted to come to. So this can be considered as a alternate or as a solution to the problem that I just posed here, right? So this can be, this is one of the solutions. So this means that here we can go for some destructive ways, although ethically it is not, you cannot do as many as we need some ethical permissions to set up experiments. Okay, we cannot simply, just because we are allowed to do that, we cannot I mean, the researchers do have some responsibilities when they work with the destructive techniques, even with the animals and plants. Okay, mostly with the animals, definitely. Another alternate that I don't know whether you have been taught these concepts or not. So this is called cell culture. Now, do you know that we have some immortal cells? Not only we, various organisms have immortal cells. And one of such immortal cells are called, the cancer cells are definitely immortal cells. They don't die. They keep dividing, they keep dividing until the individual having the cancer, once he dies, all the cells will die. Otherwise, they are kind of immortal cells. Okay. And there are other cells which have ability to divide. Okay. So, so this is called culture. Okay, cultural techniques. And there are certain there are some types of cells, they keep dividing. So if we have one such, let's say brain cells, if we get hold of it using a research technique, we can work on that, right? We can use that as a model to study it. So that is a very innovative, and that is frequently used. If when Dr. Shasikiran will talk about, he will say that he works very frequently on these systems. Okay, they are called cell lines. Okay, Anyway, I have to move on because of shortage of time. Next question. So do you recall any model organism from textbook? Let me go back to your textbook. When I was going through your textbook, I found this particular figure, and I could correlate that. If you can easily recognize that these are the experiments which Mendel had done, and this is nothing but these are the garden peas. Okay, what we call mutter. Okay, so these are garden peas, and he had done some very innovative experiments. The initial findings came from such plants. Okay, about initial finding about genetics, the principles of inheritance, it came from all these plants. But so so these can be considered as a model, but then there are better models or better alternates in biology that researchers work on. Can anyone recognize what is this? Although the size has been zoomed in, but then these are fruit flies. Everyone has seen these fruit flies. Okay, whenever you leave your food in hostel, or particularly the fruits in hostel, or if you don't eat in time, you can what will happen, these fruit flies will be attracted to that. Did you know that several genetic theories were discovered using fruit fly as a model? Amazing. You will be in higher classes, you will be amazed to know that what were those important findings that were unveiled from this. Okay, and I'm showing photo of one such legendary scientist, and his name is Thomas Morgan. You can see these bottles. That is where he used to grow them, and he used to study these fruit flies. Okay, and he was awarded Nobel Prize for his experiments. Okay, and he gave a principle of something called linkage in genetics. Okay, so right now linkage, it will be little higher for you. Linkage, you can understand that it's just ask yourself, did you notice any two traits which are co-inherited? The offsprings always have those two traits, always. That is something linkage. But anyway, it is slightly at a higher level, so I'll have to move on. Okay. But one question I want to ask you, just think about it. What is so special about these fruit flies? Why fruit flies became a model, but the garden peas, initially they were researchers used as a model, but no longer. What is so special about these fruit flies? They are too small, difficult to examine the phenotype, right? So I'm just leaving this question in your mind. Just think about it. Moving on, again fruit flies, but a different application. Several developmental process, how organisms develop, a majority of things, particularly the molecules, the proteins, the controlling proteins, the controllers, we all know about all of them, mostly from these Drosophila experiments or fruit fly experiments. So these fruit flies are amazing, and if you pursue biology, fruit flies, you have to love fruit flies. Okay, you will have to feed them fruits, then only they will come and you'll have to observe them.